Well, we're now 2 minutes past 5, so maybe we could begin, Professor.
Yes, welcome.
Hello, Elmar.
Sorry. Welcome everybody to our webinar in spring. Today is a real spring day here in Japan, with already blooming plums, trees, and warm sunshine. We have a very interesting speaker today, and we could see new people coming into our webinar, which is really giving a new venues. I can see this for Aquaphotomics, and I'm really very happy. So I would ask you again, it is always very nice to when a speaker has faces in front of him. And so it is good, and when we talk, we can talk even a discussion, etc. If we see faces, it will be very nice. Thank you, Elena. Please continue with the webinar. Yeah.
Thank you, Professor.
Good luck, Danielle. Thank you for coming.
This is a webinar. I mean, you saw the title, something novel, something different this spring. And this is really a little bit unusual webinar. It is not directly related to aquaphotomics. However, the topic that our today's speaker, Daniel Fels, is going to talk is very much of interest to us, to everyone working with water and biological organisms. And I myself have witnessed many phenomena which that might be explained by what actually Daniel is doing. So I just wanted to say how we came to an idea to invite Daniel to this webinar. When I was at the water conference in October last year, I've heard for the first time Daniel speaking about the communications between the cells, and I was absolutely blown away by what I heard. It was the best talk of that conference for me, and really like maybe one of the most interesting topics that I've listened about. It is not, you know, I mean, he's talking about communications between the cells, but in the end, all that he's saying can also be, you know, true for the way higher organisms communicate. I think you will have the same impression when you hear him talk about this. So I'm not going to waste my time and Daniel's time much longer. I just want to thank you all for coming, and I especially see many new faces, and I'm so happy, and I can't believe I'm very grateful to people from the United States who actually got up so early to be at this webinar. I'm really glad to see you. Thank you. I hope you will find this interesting. So the webinar is going to be recorded. After a few days, it will be uploaded to our YouTube channel, and it will be also with Japanese subtitles. So now I have really an honor and immense pleasure to announce Daniel Fels. Daniel, please feel free to start your talk. Thank you so much.
Thank you very much. I will share the screen first, I think.
Yes. Audience can post questions in the chat box during the talk, and after the talk we will have a discussion.
Okay. I just wanted to say good morning, everybody, but this doesn't seem to be appropriate. So just hello, everybody. And I'm greatly thankful to Professor Tsenkova, to Jelena, and also to Lawrence to invite me and give me technical support in doing so. So as you can read, I will talk about the effects of electromagnetic cell communication. But before I do this, I would like to let you know a little bit about my academic education. One moment. I actually started a master thesis working with these birds, and here I began to become interested in communication. In this case, it's begging of the little nestlings and the parents bring food. And I've just learned how perfect communication can be the parents brought the correct amount of food to allow the fledglings to grow. So that was my master thesis, and it's telling you I'm a classical biologist. I was interested in behavior, ecology, and evolutionary biology. This is where I do come from. In my PhD thesis, I worked with this aquatic small cancer, Daphnia magna, and its gut parasites, and we were testing for the manipulation hypothesis, which is very simply said, the parasites do something to the host that increases transmission success. In this case, we didn't find it, but I became interested in transmission ecology. I do not go into further details. Then I began a postdoc with Paramecia in Paris. And again, we were working with parasites, in this case, micronucleus-parasites. And we were again looking at transmission ecology. However, these cells are kind of the cells of my love. I think they are extremely beautiful. And everything you will hear, not everything, but the majority will always have to do with these cells. It's my study organism. It's called Paramecia caudatum. It's a single-celled organism. They are huge, 200 to 300 micrometers in length. They have about 2,000 cilia, which they coordinate beautifully, and they move around in water where they feed on bacteria. Now, the method that I use to find out whether these cells communicate with each other electromagnetically is a very simple method. It was not me who developed this method. It was actually Alexander Gavrilovich Gurevich, who indeed 100 years ago published the first paper on what he called cell radiation. He was working with onion root cells, and I do not go into details. What he did to be sure that the effects are not due to chemicals, he wrapped the roots into cuvettes. And this is more or less the method which we still do today. We separate organisms with a glass barrier from each other. A little detail, he found effects on cell division rates only when he used quartz cuvettes. He didn't find effect when he was using glass cuvettes. Why is this so? Quartz allows also the transmission of UV, ultraviolet light, while glass doesn't. So he concluded that the cell communication is based on UV radiation emitted from one group of cells and perceived by another group of cells. In my case, working with Paramecia, is a water organism, so I have cuvettes, small cuvettes standing in bigger cuvettes. If you look to the left, you have an arrangement of such pairs of cuvettes, with cells swimming in the outer cuvette and cells swimming in the inner cuvette. The right picture shows you 16 such pairs, which are separated optically from each other using thick carbon paper, black paper. The general hypothesis, so, on which all these studies are based, is that there is a relationship between molecules, the material part, and the fields which these molecules do release, a kind of a reciprocal causality between the fields and the matter. You can compare this with mass and gravitation. The mass has to do with the appearance of gravitation, and gravitation has an effect on mass. This is what I mean with the reciprocal system. So this simple illustration shows again what I just said regarding the last slide. Molecules, ions, chemical reactions, these all induce fields. It's unavoidable. Electromagnetic fields, they can be electrodynamic or electrostatic. And in turn, the fields have an effect back to the material component. Now, to be clear, in everything you will hear now, I will never refer to particular molecules in the cells. I just refer to the cells themselves as a whole. And I would like to start with a strange phenomenon and bring you slowly, slowly into that world of electromagnetic cell communication. What is the strange phenomenon? I observed that already during my thesis. When you repeat an experiment over time, and I think the next slide is preparing this, when you repeat, let's say every week, a particular experiment, you will see that the results vary between weeks. They vary tremendously. Even though you standardize everything, you get this tremendous variation. I was infecting water fleas with parasites, and I found effect from the parasites, which was the question I addressed. But in the statistics, the effects from repeating the experiments were much more dramatic. So there is an environmental factor that plays into our experiments, and it is not yet known what that factor is. On the other hand, it would be very surprising to do an experiment where you repeat the experiment every week and there would be no such variation. I will call this variation swinging with the environment. But what is even more surprising is if you do an experiment and you have one group swinging with the environment and the other one is not. And exactly this is what I found in some of my experiments. And this is how I bring you into that world. I had experiments where when one group was influencing the other group, the influencer, it's called IP here on the table, and the receiving group tester population, the inducer population did not anymore be dependent on that external factor. I have no answer to this. I do not know why it stops. I even do not know what that environmental factor is. But I found it in two different experiments. In another experiment where I had one pair of cuvettes with an induction and another pair without induction, I found this. In the table you see under 1A quartz separation, you see that to the very right, the tester population still showed these effects due to the repetition of the experiment. Every week a repetition, 3, 4, 5, 6, 7 repetitions. There was this tremendous effect just from repeating the experiment. When you look to the left in the first line of data, this is the inducer population. There was no effect from repeating. And in the middle section, you see that the inducers had a significant effect on the tester population. I will later speak about these effects. Allow me just to say there were effects. And we look at these swinging or not swinging for a moment. In the lower line, the glass separation, no population had an effect on the other one, and both were swinging with the environment. To me, this is still very interesting, and I know that some researchers believe it has to do with cosmic radiation. That's the inducer. Others assume changes in the Earth's magnetic field. I'm not going into this direction. I just wanted to bring you slowly into that unknown world of electromagnetic cell communication. And I think with finalizing with that picture, this just repeats what I said. If one population has an effect on the other one, the inducer stops swinging. If not, both populations swing with the environment. I could publish this work 10 years ago, 11 years ago. It's, if you want to read it, it's quite an effort to delve into these thoughts. It has to do with entanglement as well. But I skip and would like to bring you now really into electromagnetic cell communication only. Opposed to the Govich experiments, I found effects also across glass, not only across quartz. I also saw that the effects did not only induce cell division, but also decrease cell division. And I also realized that depending on the neighbors, cells were eating more or eating less. So there are diverse effects, not only cell division rates that are enhanced, which is why I call it the effects are more than mitogenetic. Here we go now a little bit into first data. This is one of my first experiments where I only addressed one question. Is there an effect from neighbors? Yes or no? Nothing else than that. And I was using quartz separation and glass separation. The black squares refer to glass separation, the open squares to quartz separation. To the right, you have two types of controls, which I do not mention here. You can read it in the paper, but two solid controls. And what it told me is that there are effects across quartz, there are effects across glass. Stunningly, in one group, the bigger population of 100 cells was influencing the growth of 5 cells. And in the other type of separation, the 5 cells inside were having an effect on the 100 cells outside. I do not interpret this here. I just wanted to show you these data because I made many repetitions for these results, 14, 16, 17, 18, I don't remember exactly, but very many repetitions because I wanted to be sure there are effects. And this is another experiment where I was interested in how much do they eat. For that, I have to kill the cells, fixate them, and just count how many feeding vacuoles do appear within the cells. And the result here is that first, when the cells had many neighbors, 300 to 400, or only a few neighbors, 15 or 20, they were eating either much more or much less. They were eating much more with many neighbors in case of glass separation, and they were eating much less in case of quartz separation. Still, I do not interpret it. Please note, I wanted to be sure, are there effects at all? This was a double-blind study. And I became convinced that there is really something to it and could publish it. This is the most cited paper, but in my opinion, not the most interesting one. Now, I became interested, what are they talking about? What is it? What is the meaning of it? What is the function? Is there a function at all? And in order to find out about the function, I was only looking at quartz separation, the results of quartz separation. Why? Under natural conditions, there is no barrier between the cells. So whatever photons, whatever electromagnetic waves they do emit, all of them may potentially reach another neighbor cell. Now, with quartz separation, there is a maximum of electromagnetic waves that can trespass from one group to the other. So I was only looking at quartz separation. And then there was a common a common phenomenon. With many neighbors, there was a decrease in cell division rate on the side of the tester population. So the more neighbors, the less they would grow. And this was interesting. Why? When you maintain Paramecia in a falconer tube, in a micro ecosystem, they reach a total number of about 300 cells, not more. But these 300 cells occupy about 1,000th of the water volume. There would be much more place for them. And food is also abundant, but they do not do it. It's not difficult. 3 hours later, they could be 600 cells or 1,000, 2,000, 4,000, but they do not do it. So how is that density maintained? There has to be a certain communication going on. And I wondered whether it's electromagnetically regulated. And for this, I made the following experiment, which I show you now. Have first a look. The design of the experiment is presented in the above line, where you see these cuvettes from top. In the outer cuvette, which I call the inducer population, I placed either 50, 100, 200, or 300 cells, knowing the 300 cells would not grow during the 2 days of exposure of smaller and bigger populations. During these 2 days, they would not grow, while the 50 cells would grow. Now, if growing or not growing is electromagnetically regulated, if this is the case, then the electromagnetic waves would also trespass from the outer population across the glass barrier to the little population inside, the 5 cells inside. And I assumed that the 5 cells inside would grow differently, in dependence to the number of inducer cells. And here are the pictures. Please look first to the left graph. The left graph is simply what I already knew. 300 cells, look at the x-axis to the very right. The 300 cells would not grow anymore, while the 50 cells to the left, top, they still had a strong growth, cell division rate. Now, when you look to the right, this is the growth of the 5 cells inside. All of them were growing, but those that were surrounded by 300 cells grew much less than those that were surrounded by 50 cells. And this is the first result now where I became aware that there is a function. It's not just a by chance effect, and one function could be regulation. And not only just the induction of cell division. Now, why do I underline the term regulation? I will come back now again to the Gurvich experiment. These are very old pictures, 100 years old. And to the very left, there is a scheme of an onion root emitting light to a detector root. And within that cylinder of the detector root, you have real pictures of mitosis, cells that proceed, that are mitoting, so to say, yeah? Also, the nucleus is divided into 2 nuclei. And as you can see, on the left side in that square is more black, means there's more mitosis going on than on the right side, which is why Gurvich concluded that there is an induction in cell division rate due to cell radiation. But Reiter and Gabor, they were 3 years financed by Siemens in the 20s. Siemens thought probably we can produce some technical devices for a non-invasive therapy. However, coming back to Reiter and Garbo, they repeated the Gurwitch experiments, and they did not only find that on the exposed sites, cell growth here was enhanced. They also realized that on the other side, there was less cell division going on than on further top or further down regions, which is why they said it is regulating the total amount of cell division and not just increasing. And if you look from an ecological point of view, we could also say this, that if here is a root growing, then this root grows away. So there is no competition. And exactly growing away is the result when here are more cell divisions than on the other side. Then it grows exactly into this direction. But let's keep this in bracket. The term I want to leave you with here is regulatory function. I believe that electromagnetic cell communication has a lot to do with regulation, but this demands further research. I go on now. More details about other studies will be published soon in a book with the title Ultraweak Photon Emission from Biological Systems. And I was invited to write one chapter. And in that chapter, I focused on the possible functions of electromagnetic cell communication. In that chapter, you will find many examples of effects induced by electrical fields coming from cells. Now, in those days when electromagnetic cell communication was discovered, people also made experiments across the species border. In that chapter I just mentioned previously, you will find such examples. I was interested whether I can also find such effects. And I was looking at effects coming from Euglena to the left, a unicellular algae, very small one. I could keep in 1 milliliter 100,000 of these cells. And in the neighboring cells, I had Paramecia. And there was just the effect without explanation that the growth in Paramecia was reduced when they had neighbors, Euglena, as neighbors in this case. It just hints on the possibility that in complex ecosystems, the total number of species, the species composition, might also be under the influence of the electromagnetic waves emitted by all of them. I did another experiment, again testing for effects across the species border. To the left, you see Rotatoria. It consists in about 1,000 cells. It's only a bit bigger than Paramecium. I told you Paramecium is a huge cell. And I just wanted to know whether they affect each other. There's one thing I did not tell you. Usually, when I make such an experiment, and I have these pairs of cuvettes, I place them in a black box and allow them to grow during a period of 2 days, 48 hours. After 48 hours, I open the box and I count the number of cells. Very simple method. I did the same in this case. I was counting how many Rotatoria did I take out as compared to how many I placed in. And the same I did with Paramecia. And there were significant effects from one to the other in both directions. And if you look now at this quite complex figure, please let me guide you through that figure. It summarizes the results of between species effects. The lower part is a kind of a repetition of the upper results in the right panel. Don't waste your eyes by looking at these figures. I start with the panel C to the very right. all these effects, these bars, these were all significant effects, just showing rotatoria can influence Paramecia, but the next week they do not. But then Paramecia influence rotatoria, and it depends whether they were separated by glass or by quartz. I cannot explain these results, but again, please be aware, it's absolute basic research. So the beginning is just, are there effects at all? In this case, are there effects even across the species border, which hints to the possibility that we look at a universal phenomenon, not only at a species-specific phenomenon or a species-specific language. It might be a universal language that life uses to regulate itself within species and between species. Please look now at panel B. Panel B shows you the experiment with euglena, the photosynthetic little cells on Paramecium. Why do I want you to look at this picture? This is because in that experiment, I also used graphite to make the inner cuvette black. By that, no light can trespass from the outer population to the inner population. This is an indirect testing for electromagnetic signals responsible for observed effects. And in this panel B, the two mean values and standard deviations to the right, they show you growth of Paramecia in the control condition, no euglena outside, just medium. And the other one shows you euglena outside, yes, but a graphite layer that doesn't allow electromagnetic waves to transmit. To the left, no graphite layer, the test itself, growth in Paramecia was lower when they had euglena as neighbors. Now, what is the outlook? The outlook for me is future experiments I would love to do. And here you may have a look to the right picture first, please. Here, there was a site observation. I did not directly test for it. It was a posteriori. And I do not go too much into details, but what do we see here is the following. When I place 300 cells at a maximum density in the outer cuvette, you remember they would not grow. But it happened that some of them showed a strong mortality. Means I placed 300 cells into the cuvette, but 2 days later, I was counting 220 cells or 250 cells. So when a population remains stable, you have cell division as well as mortality. This is how a population remains stable in numbers. Yeah, cell division and mortality are in balance. Here, there were cases where this balance was not there. The inducer populations at high density became smaller, so there was cell death going on. And now the strange thing is that the more cell death there was, the more the inner cells grew, which I'm tempted to call this compensation for cell death, or even death compensation. But here, I will have to test directly for it. This is one of the experiments I would like to do in the future. To the left, you will see 2 bars. And in that experiment, I just wanted to know whether 100 cells also have an effect on a single cell. Because the 5 cells were already communicating with each other. So kind of excluding that, just 1 cell surrounded by neighbors. Now the general trend is if there is no mortality in the outer cell population, if there's no mortality, if they grow or remain stable, the general observation is that the cell in the inner cuvette would grow less. That's a general observation. But in this case, the single cells grew better when they had neighbors. So this is probably not contradictory. I'm interested in what are they talking about? What kind of language, system, organization, principle has evolved over millions of years within these cells? And I have yet almost no clue, just a little bit. So we are at the very beginning scratching a little bit into that world. If you look now at the left panel and to the right of the left panel, you have symbolized 3 cuvettes. And this is an experiment I also would like to do. Imagine now that I have 3 cuvettes together, not one standing in the other one, but 3 of them together. The 100 usually have an effect on 5 cells by decreasing the 5 cells. But the 100 also had the effect by increasing the growth of a single cell. The single cell made cell divisions. Now, when I bring the 3 together, my question is, do the 100 have effects on both the 5, decreasing their growth, and the 1 increasing its growth? This would mean that the cells interpret the signal for themselves, or do the 100s make a decision and say, let's say that a little bit funny, the 5 are already 5, they can do their own thing, but the single one needs some support. So is there a decision-making process going on? To me, this is an interesting question to delve deeper into that world. Yet, these experiments are preliminary data, not yet further tested for it. This is something for the future. In conclusion, summarizing the above studies and others, I conclude that mitogenetic radiation is much more than it was assumed to be 100 years ago. It is actually, it has a universal regulatory function, this cell radiation. And there's still not much we know about it, but you could also say there's still a lot we can learn about it. And coming now slowly towards the end, I would like to emphasize this strongly. Here is a picture from a kind of an essay. The essay had the title, "The Double Aspect of Life." And by the double aspect, I think you all realized it now. I refer to the material part of life, all its molecules, and to the fields that appear due to electric charges, chemical reactions, ions, all of these cellular processes. If you look at this panel to picture A, I just want to say all cells perceive material, they are eating food, and sun rays and other kind of rays. This is normal, and photosynthesis is the fantastic result of this perception of electromagnetic rays coming from our neighbor star, the sun. Panel B is what we have been looking at now, not the exchange of material. Parasite transmission is a kind of an exchange of material, you can even put it like that, but on the potential for electromagnetic cell communication. These wavy lines want to refer to that. Now, if you look at C, this is also interesting because these electromagnetic components of a cell, they have the potential to play a role in the process of evolution. Why? These electromagnetic waves, there are not all of them not all of them are emitted by the cells. They also stay within the cells. There is physics that can explain such a phenomenon. Resonance is the key word here. And so if you imagine an egg that is fertilized by a sperm, the egg brings an orchestra of electromagnetic waves into the next generation. And when we think of evolution, it's always the transfer of existing material, existing fields into the next generation, and changes in the field composition and in the material composition. But studies to look at even the evolution of electromagnetic fields, this is again something for the future. There is now a second panel, which you will find in that book chapter, in the book that will release this year by Springer Nature. And I just want to make aware with that picture that we can look at very many different systems to learn more about the existence of these fields. And maybe you just go by yourself a little bit into that picture. On the lowest depicts a cell, and to find out within a cell what the functions are of electromagnetic waves, this could be quite difficult. We would need nanosatellites, but such satellites already do exist. We can imagine to look at the conversation between parasites and hosts, symbionts and hosts, mycorrhiza and roots of trees. More than 90% of all plants live together with fungi in the Earth system. There might also be an electromagnetic regulation going on. So I just was showing you a little bit of that vast potential total field now for scientists, field to discover for scientists. I only showed you a little bit about how cells can affect each other across glass barriers. So with that little picture, I'm coming to an end. You see two birds, one doesn't allow the other one to talk. I think maybe some partnerships suffer from similar problems. This is a joke. This is a joke. But instead, please replace male and female by molecules and fields. The fields have something to say, and I wish the community of all life scientists just to look at them at least, do not deny that they can have a tremendous influence. Some people go very far, and they say the majority of phenomena we have observed are probably wrongly interpreted because fields were co-factors in all of the majority of these experiments, but nobody looked at them. I don't want to go that far. I don't want to attack. I just long for a union of molecular biologists with, let's call them electric biologists. I thank you all for your attention and leave you with these two Paramecia in a beautiful relationship, whatever they do. Thank you, all of you, for listening.
Thank you, Daniel. Oh, I really hate that whenever we have Zoom, I cannot give, applause, loud applause, but I mean, I can only do this for you, but this was really wonderful. I'm so grateful, and I think the other people are grateful too for this wonderful talk. And your talk already sparked many questions. So are you open for questions? Can you start now, or you need a little break? Definitely.
I drink a little bit of water and then you start.
Wonderful. So, okay, whoever has a question, please, you can just raise your hand, but I will start with the questions that we got in the chat box. So the first one was by Robert Alfano. Actually, yep. Robert, if you want, you can,
yes, I have a question.
Please, yes.
I have a few questions. Very nice presentation. I'm very interested in cell-cell communication. I am not a biologist, I'm an optical physicist, and I'm involved in what goes on in materials. And the question I have for you, I have two questions. One has to do with, did you ever block the cells with a metal between cells or a piece of cardboard between cells to see what the effect is of the electromagnetic radiation between the cells? Because you could terminate, obviously, with a piece of metal, all the electromagnetic spectrum that is shielded by the phenomena that you see. The second question is related, is what frequency of the electromagnetic spectrum is responsible for the communication? Now, this reminds me when I was a student, I was studying Froelich, which you have, I think, in your book, in which he talked about life at low frequencies, and it was very important of excitons and all these general phenomena. So the question is, what frequency domain do you that does the communication? So that they also, all the electromagnetic radiation is at the speed of light. So you got low frequency radiation, you got RF, you got near infrared, you got optical, and then you got UV. You've been focusing on UV, but that's a very small portion of the electromagnetic spectrum. So do you have any idea of what zone are we talking about? And then once you figure that out, then it goes back one more step. So all this information that you generated, which is wonderful, is the fact that all the bases of cancer, for example, may be based on electromagnetic radiation that you said. That cancer, the cells are communicating, and the tumor cells communicate more or less than a normal tissue. So, and also goes back, you said also, you hinted at photosynthesis. That's based on light. Without light, you don't have photosynthesis. And that's basic of light. We wouldn't be able to breathe. So all these fundamentals that you talk about, blocking the cells from one communication to another, the fact that you have possibly what are the underlying process, like in microtubules, there's this radiation they see that doesn't come from anywhere, but it comes from the cells of microtubules, and the microtubules may communicate that information in the brain. So this is all fundamentals in the sense of what's going on in your idea and methods. You've been doing this for years on communication between. I hate to take such a long time to explain some of the basics, but that's what I do. I do a lot of optical phenomena. My career is based on that. So I should say I'm from City College in New York. And I talk like I am from New York, as you can tell. It's my okay. Very nice to meet you, Daniel, for the first time, and hopefully in the future we'll meet and discuss this in more depth. But I appreciate this society, this aquaphotomics. I'm very big on water. Water is very important, but that's a separate issue. So I just got these fundamental questions. What's the effect of the shield? If you put a metal, what frequency domain we're talking about? And also how this affects cancer or uses communication?
I will answer you one by one.
Okay.
Metallic shields were used by Albrecht Bühler 20-30 years ago. And in that book chapter, you will find references to it. When he was shielding, he did not find any more effects. In my case, I will show you this. This stands now for a cuvette without shielding. And this is my coffee cup, you know, which is black. What I did, the shielding I did was that I used a graphite spray and I sprayed the inner cuvette, its outside wall. Then I placed it to the bigger one and there were no more effects. That's all I did regarding shielding. But it is kind of it's kind of convincing that it could be electromagnetic waves or photons. Now your second question. As in cells, there is a lot of infrared going on, warmth, just temperature, just simply temperature. Any electromagnetic signal that is of higher energy has a better noise to signal ratio. So just theoretically, cells should use visible light, photons in the visible range, and even in UV. And in fact, measurements show that cells do emit in the visible, the whole range of visible, also in UV. And your last question regarding cancer.
Yes, communication and cancer.
There were groups, and probably still are, that were measuring the amount of photons emitted by malign cells as compared to healthy cells. So isolated malign cells, isolated healthy cells of the same cell type. And the cancer cell released much more photons. However, the theory behind is quite complicated, and I would have to reread all of it. I cannot give you more of a detailed answer regarding cancer.
Okay, thank you. Very good. Very nice.
Thank you, Robert. Very interesting questions, actually. I would always bet that the range of frequencies used is near infrared and infrared, based on what we see with what is happening to water and how this range of frequencies affects the water. But I cannot exclude others. So there were many comments on the questions that Robert asked coming from Pierre. So Pierre, would you please be kind to turn on your mic and say at least some of your comments?
Can you hear me?
Yes, okay. Please.
Hello to everybody. So even though the time zone and time shift is a little bit hard for someone to attend, but anyway, I'm glad we could make it. Yes, Daniel, it's quite a challenge. I think you should do a half-an-hour talk and a 2-hour discussion, because what you triggered off again is an avalanche of inspiration and ideas. And these ideas, I think, have huge implications of what we are currently living and going through, particularly if you look back at the 2 years of pandemic. Because I think if this electromagnetic coupling is a reality, then you have to do with matter, Paramecia. Then we have to do with the energy transformation, which means energy is involved in the biochemistry, in everything that the metabolic activity of this organism does. And then you have the information aspect. So we have a triadic system, and where information, matter, and energy are intrinsically interwoven. So the idea is, how can we allocate what is the relevance of these three, let's say, entities? And one aspect would be, okay, what happens if you remove the inducer population, just leaving the aqueous phase there, and see what happens to the tester population? Which means if there is electromagnetic radiation taking place, and this would fit to Yelena's introductory comment that, well, where's the bridge to aquaphotomics? And here, if we have an the organism, then it should be prevalent also in the aqueous phase, and you should see somehow in the changed infrared spectrum in the near infrared region, which means okay.
Pierre,
Go ahead.
Can I directly reply to this? In one of my first slides, you could see effects across glass and quartz, and on the x-axis there was written C1 and C2. These were two types of controls, yeah. And but actually, these are not the kind of controls you would love to see. You would love to use as an inducer population the water wherein cells lived for 2 days. Then I remove them all out correctly and see whether there is still an effect. This is highly interesting. This is highly interesting. It's very beautiful. I never have thought to do that. Yeah. So there is nothing I can say about it. And I mean, it would be unbelievable, so more or less, yeah?
Well, you can refine that, Daniel. You can even refine that. And what happens if you dilute this, make a serial dilution? And if Montagnier is right, and he had documented this effect on his EGE components, then I think you should see a shift in the dilution series when you have a diluted, let's say the aqueous phase is diluted, and then the I of the information matter energy triad becomes so relevant that at a certain dilution, you obviously, or that you might see so strong effects that probably you have to rewrite or induce a paradigm shift in a biological understanding. Because if that's really taking place, we no longer can fit with the biochemistry model anymore.
You're absolutely right. I mean, you know, sometimes I think what I'm doing is already science fiction. And what you're talking about is the science fiction of science fiction.
Well,
no,
I'm
not absolutely not.
Absolutely
I'm not so
not.
sure if it's science fiction, because Elmer Fuchs.
No, I mean, by science fiction, I just mean work for the future. It's avant-garde.
It is for the future. Yes.
And,
yes.
And this is avant-garde of avant-garde, which doesn't mean I do not think it's interesting and that we should do it. Please understand correctly.
It sounds like Leonard McCoy when he's sitting on Star Trek in his first aid medical recovery room, no? And he's using a scanner, and he's
exactly.
And the patient, and it shifted, completely shifted. So what happens if you use a technical field in the ELF range and expose it, such as a coil, put the tested population inside, and see if there's a reproduction happening, scanning through the frequencies. And you start off with that frequency that Mark Henry wrote in his paper, in his legendary paper. I forgot the name, but he uses 50.4 hertz and finely tuned. It's not 50.4 hertz, but it's 50.400 hertz. So the significant numbers are very important because it's so finely tuned. And suddenly, there's so many things happen. We have done an experiment with George, and this was mind-staggering. But this is a little bit out of the scope. So, but I mean, this would actually be the next one. And that's what Elmer Fuchs, in one of the comments in the chat box, said, what is the modulation? Exactly. What is the modulation? How it is modulated? And if you have UV as a carrier, it's not just UV, it's frequency modulated, amplitude modulated, quadratic modulation, phase shift modulation. What is it?
Look, I cannot give answers to this, you know. For this, you need engineers, physicians, physicists, yeah, you know, who can do these experiments. I'm just taking the whole cells as an emitting unit and the same as a receiving unit. You know, these are the details you have to do in the future. Even the machinery to make all those measurements you're longing for and asking for, I cannot provide that. But if it inspires,
I mean, we can provide that kind of instrument.
Okay.
Sorry, I had to just jump in. Absolutely.
Yeah, do that.
Yes.
However, a little thing I would like to refer to one of your demands for future experiments.
Yes.
Reiter and Gabor in the 20s, they found out which frequencies the onion roots are using. And then they developed a quecksilver lamp that was emitting exactly that frequency. And they emitted that frequency to the cells and they got the effect. Now, wait a minute, interesting. They found two frequencies and they applied both frequencies, but not together. Either one or the other, they found these effects. However, when they applied both frequencies together at the same time, there was no effect. So there's still a lot to learn about these systems. I think our way of thinking and understanding life will demand a lot of intelligence in the future, but we have it.
Great, Daniel. A final comment.
Please.
Then I stop because the others want also to say something. How about if I send our testing equipment into your lab? In this case, I mean the PMT tube to get the measurements done.
Yeah, Pierre, I have now my answer is a very private one, okay? In a few years ago, I had some dramatic changes in my private life. And I'm right now in a phase of my life where I don't have the energy to do the time and energy to do such experiments. I have to wait for my retirement. It's very sad. I'm very sorry to let you know. I'm extremely curious about all those future results. And I told you which experiments I would love to do. And as long as I can be inspiring for other research groups to use their methods, their technical stuff, their ideas, their understanding, to delve into it, I'm so happy. And I will always be ready to talk with you, share ideas, no doubt. But unfortunately, I do not have the lab with 12 co-workers and 70 hours time a week. I'm sorry for that, Pierre. But maybe I can send you my cells and then you do it in your lab with your material, you know? That's what I can propose.
Okay, let's talk about it later.
We will try at least here in our lab to do something about these experiments.
Fantastic.
I'm sorry, so Pierre, sorry, I have to cut now this because we have questions from other people, although I would so like to comment on many of the things that you two now exchanged. But I would be, you know, like act as a lady and I will give opportunity to other people. So there is a question from Mr. Shei Xing Sheo. Would you like to just, you know, turn on? Thank you. So you can ask, please.
Yes.
Thank you so much.
I'm from Thomas Jefferson University at Philadelphia. So it's like 5 o'clock in the morning here. Okay. So I have two short questions. I work on mitochondria in heart. But the first question is, this cell-cell communication, if it occurs, say, in heart, the heart is the one which is constantly firing the action potential and they are electrically connected. So how do one apply your concept in terms of electromagnetic communication to an organ like heart that I would think generate the most electromagnetic field because of their intrinsic electrical properties?
Okay, I would like to repeat your question to be sure that I get the question as you addressed it to me. You think that if we are looking at organs, the total amount of electromagnetic waves that are emitted should be dramatically bigger than the amount of electromagnetic waves emitted by a small group of cells. Is that correct? Is that the basic?
Yeah, and then how, if that is the case, how that apply to the control, in this case, the heart function?
Oh, so actually these are two questions. I don't want to escape the answer, but I have to be humble. I'm just not saying more than I can. And I will approach this from another side before I come to that. You know, what I think is amazing is our body consists in about 38 billion cells, now European billions, so American trillions, which is a tremendous amount. And all of them are our co-workers. And what to me is just a joke of life is that I anyway have the feeling to be one, even though I consist in very, very many. And I wondered, how are the cells able to give me a feeling of one? And one answer I do give for myself, this is philosophy, this is not any more science, in a sense I cannot test for it. But if you imagine you have a little magnet with its fields, and you add a second magnet, then you get one bigger field. And if you add a third one, you get an even bigger field, but it's only one field. And sometimes I have the feeling that there is a total of electromagnetic fields in the whole body that allows unification of all these cells to be, as I said, kind of one. Now, if we look into this kind of a holistic perception, then there are minute processes going on, like cell growth regulation. We never look monstrous. The number of cell deaths and replacement from stem cells is obviously very fine-tuned, and there could be, the reason for that could be an electromagnetic regulation within the body. Now, coming to the heart, whether the electromagnetic fields play a role in the function of the heart, there might be two functions of the heart. One is just the pumping of blood, which is organized by muscular cells, the heart muscle cells. And there we just have the action potential explanations, nerve cells touching muscles, acetylcholine is released. But this is rather the electrostatic phenomenon, the polarity changing on the membrane surfaces of cells is less what I was talking about, photon release. So of this long answer, actually, I cannot answer your question. I wouldn't know how, but I think it's very interesting.
Yeah, just quickly follow that up. Does the electromagnetic field in the cell that you are working on coming from the organelle, like mitochondria, which has the most negative membrane potential? And,
Yes.
and if they do oscillate, they would generate a field. What do you think? What's the origin of the field coming from?
Okay, yeah, thank you for that question. I mean, there are several origins, but most important are ROS reactions, reactive oxygen reactions, and you find them around mitochondria. So the sources are oxidation processes occurring within cells where the educt and the product have different energy contents, and the energy difference is released by light. For that, there are quite a few papers from friends in Czech Republic. They do publish this. There's a nice group. And if you go into my papers, or also the book Fields of the Cell, it's not the one that was presented here on the webinar site, but it's a book with several chapters. Pierre also contributed in that book. There you will find references, and in later papers of mine, you will find references to people that want to know, where does the light come from within the cell? Where does it come from? I hope this was a little bit of a help. There is an understanding of that. But again, I'm just looking at cells. I want to understand what has one to do with the other.
Okay, finally, you are welcome to come to my lab to do your sabbatical. We have a beautiful lab, and we have what we call a Mito Care Center. You can Google that at the Thomas Jefferson. We have one of the most, I think, advanced facility in terms of studying the mitochondria. So you are welcome in your spare time to come to Philadelphia to do experiment. Thank you so much. Yeah.
Thank you, Professor. Wow, this was really beautiful.
Yes.
So, yes, of course.
I just want to add something, because it's such a nice conversation. We, at the moment, I was listening to Daniel, and usually what happened is you concentrate on the cell, of course, on mitochondria. This is something that we have to know and follow, and we know. But we exclude the water surrounding these species. So they live in that water, and the water is the media, is the field as well. So we definitely, and what is happening now in all these webinars and in the research that we do also, is getting all these two fields together. And absolutely. I think this is what is going to produce the answer. Because I've been thinking all the time, listening to you, about coherence. and Antonella is here, so maybe she can add something.
Yeah, that would be nice. Yeah.
Because all those frequencies and the presence of some of other cells, it is coherence, and the energy that comes from that coherence is the key. This is what I think, and what we have been seeing already in our experiments. So, and this is the future. So and this is very good that we have also labs working with mitochondria, and probably can apply aquaphotomics as well. Thank you, Daniel. It was absolutely fascinating.
Thank you, Professor Tekla.
Thank you.
So you mentioned Professor Danilo, and Professor Danilo actually had a question in chat box. So Professor, can you just say it aloud so people can hear? She had a question about the noise, actually, if introducing. Please.
I was wondering why, even if it's so difficult to find the specific frequency of the communication, why don't do the opposite? I mean, to inhibit the communication, just saturating the environment with electromagnetic noise in order to stop any kind of communication. This is much easier.
Absolutely.
Yeah. That's a fantastic idea. That's a fantastic idea. But what would the result? What would it tell us? Would it tell us more than if there is an electromagnetic conversation going on, it can be disturbed by a tremendous amount of electromagnetic noise? This is like, I mean, why not doing that? There is another idea which I'm talking about with Felix, that we use optical fibers and we connect two cuvettes. So the only signal that can transmit then across an optical fiber would be a photon. But this would just answer the question, are these really photons that are responsible for the effect? Of course, even though we cannot think of anything else, yeah, it is still not shown directly, because to show it directly, I would need to catch a photon that is emitted out of the cuvette, store it, and place it then back to another population. Such a thing you can do with hormones. You can take thyroxine of a baby frog or an old frog and send it to Canada, and they may place the photons to baby frogs, tadpoles, and they will start metamorphosis. This is beautiful, but exactly the same testing is not possible with the photons. So I love your idea that the disturbance could disturb the phenomenon, but then we still wouldn't know what they are talking about. But it's another question. You see, my question is, what are you talking about? I want to sneak into the world of paramecium. I'm less interested, even though it's interesting. It's interesting to know how the mitochondria might contribute, how the microtubuli of the cytoskeleton, how they might contribute. This is all very interesting, and I do not deny that, but it's not directly what I'm asking. Yeah, I'm a biologist who kind of studies the behavior. You know, if you hear birds singing together, you might be interested in how do they produce the sound, or you just want to know what are the effects of singing, pairing in the end. Yeah, and I'm looking on that level. Please understand that I'm not denying the beauty of your thought with that.
So you could ask the question, what code is used in the communication? Because you have the random noise and you have now, what is the communication code that they communicate between cells?
Can I try to answer that question if possible? I think
Please.
it's just light of different frequencies.
Yeah.
Different frequency means has different information. So for example, something that we saw from papers and our experiments, if you use, for example, if you want to count bacteria based on the spectra, you know that the information on the number of bacteria will be at the one absorbance band in particular, which is 810 nanometers. And the interesting thing that I found from the papers, for example, is you want to increase the number of bacteria, if you want to make them proliferate more, you can apply a laser with this exact light wavelength, and that will induce proliferation. So it tells me that this particular frequency of light contains information that, like says what will be the number. It is definitely, connected to the number of cells. So in the similar way, I think there are other frequencies connected to the other information. And what I wanted actually to hear, Ed, we did not, I really loved how Daniel noticed, and he mentioned so often this regulatory function. And this regulatory function, I think, comes exactly from, it has to be connected to the information present in the system, right? Because in order to regulate something, you need to know how, right? So, and we exactly in aquaphotomics, when we talk about what we see in water, we have, let's say, three let's say, parts of the water, which we usually call, Professor Tenko invented those terms, working water or working horse, the battery. And there is one very, very small part, which we absolutely know governs the behavior of these other parts, and we call that the regulator. And we also had this regulator part of water. It is like a very, very small part of the spectra of around 10 nanometers wide range. And we had found that for the amplification of DNA in the experiments that we did with late Professor Luc Montagnier, we found that this regulatory part of water, regulator, was crucial for successful amplification of DNA. So it has to be connected to the transmission of information, to the information itself. So this means that the water also is absolutely crucial for this process. And I think it works as a sort of, like a radio. It catches the frequency of light. So it basically works like a radio station, something like that. So I'm sorry, I know I talked too long, and now we have also some other questions, but I really needed to say that. Thank you for listening. So we have more questions. Actually, these are comments from Professor Denino and Pierre.
Can I also say something? Do you know about the 4 optical windows in the near infrared of imaging in water? Water is got 4 optical windows. I designate these windows 1, 2, 3, and 4. These are the valleys of absorption, and there's these mountains. I don't know if how many of this group understands the transmission in the near infrared, starting from about 800 nanometers. You mentioned the word. That's window number 1, and then it goes into other windows, which allows deeper imaging in tissue.
Yes. Based, of course. Well, we usually talk about the overtones of water in this near infrared, but what you're talking about is related to the penetration depth of light into the tissues, right? Yes. That's correct.
That's correct.
Yeah.
And then there is windows where there's other molecules, like in window number 3, which is at 1700 nanometers. It's got collagen there. And so depending on how you're going to probe a tissue with light and image it, you have to pick one of these near infrared windows or WIR windows. And I don't know if anybody knows about those in this group, but that's a very important, and this was done by my postdoc, Binyang Shi, who's now a professor at San Diego. And she does fantastic work of deep imaging of the brain. And you should invite her to talk.
Well, wonderful idea. Can you just give us her contact? I think I might know the paper of your postdoc that you mentioned, if by any chance. So it was very interesting for me because one, for example, all these publications that are coming, I mean, for me now they're becoming very, very interesting from the area of biomodulation, using the light to stimulate some effects or induce some effects. So that for me now provides more information about, because we see, for example, the same bands that we know correspond to, for example, absorption of particular water species. And we see people using the light of those frequencies to induce some effects. So everything that is used, you know, like I think maybe one of the papers I read that the laser which used light at the wavelength 1742 nanometer was able to enhance the communication between neurons. And that was connected with the change in the water structure that was detected by exactly near-infrared imaging.
The absorption peaks I'm talking about are doing combination vibrational modes of water. So there are three frequencies, vibrational frequencies of water: nu 1, nu 2, nu 3, and depending on the combination, you get near-infrared, these absorptions from the vibrations that exist in the tissue. So
well, yes, well, in aquaphotomics, we actually go in more details, even more details. There are these large overtones, but within these overtones, there are particular absorbance bands. So we make it more precise somehow. How can I explain? So that is what we do. Sorry.
Sorry.
Any more questions? Anyone?
No,
comment more?
Pierre wrote a very nice note about extraterrestrial influence on biota. I think so. We don't have Vladimir or Elmer to talk about this, but this is something that definitely what Daniel mentioned, the environment. Okay, what is the environment? What do we have in that environment? And if we have the hypothesis that we have this water structures everywhere, then that might help also. Because this is something, for example, in all our experiments, usually we have observed a beautiful correlation with the time in any system. Because we can do monitoring of that system in real time, and then we do a regression against the time. So it means that we see the dynamical changes that are in high correlation with time. So now the question, what time is?
Yeah, I want to add on that. When I was listening to Daniel's talk at the water conference, what he was showing about these populations of cells swinging with the environment, I was like, oh, that is how water behaves. This is something we see, you know, because we monitor the spectrum of water over time, every day. And we observe this, that every week it is a little bit different, you know, like it's changing. So I was like, it's not only in the cells, it's not just the living organism, the water is actually catching all that. Oh, okay. Thank you, Pierre.
I would like to add to this. You know, I heard of a Russian scientist who over months, very many months, over a very long period, he repeated every day the same chemical reaction, A plus B, looking at the amount of C. And he also realized that he does not get the same quantity of the product. So there is something going on. I think that influences all matter, not only life. And there is something else I would like to add. You know, sometimes when we talk to each other coming from different schools of thought, we may not have the same definition behind a word we use. I realized when I said communication, some people understood differently They did not understand how I define communication. But this is not the point here. The point here is another one. The dimension as I'm looking at is the micrometer dimension of whole cells. The moment you want to know where from does the light come, we go into a dimension that is 1,000 times to 1,000,000 times smaller. It's like not another world, but just for all of you to understand probably better, you know, I'm in a, oh, this sounds pretentious. I'm not in a higher dimension, please. But in, yeah, how to say, yeah, I'm kind of in the micrometer world, yeah, while you are in the nanometer world, and there you ask different questions, of course, you know, but these I cannot answer, yeah. Imagine I'm watching just tigers in the forest. This is the level I am, while you are deeply within cells and have other methods to do. So this is probably also why I cannot match the questions you are addressing as close as you wish them to be. However, there's a little story with water I would like to mention. I have aquaria, a small one with 30 liter and a bigger one with 140 liter. And there are some snails. And the big aquarium didn't have snails so far. So I took snails from the smaller to the bigger. Now here comes the point. These snails, they can be small or bigger. If they are very big, the shell has a diameter of 1 centimeter or 12, 13 millimeters. In the small aquarium, they all remain small. You never get these big snails. I took 2 small ones. And within a few days, both of them were very big and copulated like crazy the whole day whenever I saw them. How did they know that they are now in a bigger aquarium where there is more place so that they could grow, build bigger ovaries, higher sperm production, and just reproduce like crazy? And to me, the answer is that the water is telling them. The water is telling them also how much it is in that aquarium. And this is perceived by the snails. That was the only logic answer I could find for myself. There might be others, molecule-based, the smell of smell and so. So this is
this is so beautiful what you said. I love your story. For example, recently I found out there are a few water absorbance bands which contain the information about the volume of
really
system. Yes.
This is amazing.
It was amazing. I was also, you know, like
water is alive, kind of. Water honestly, is alive.
Yes, honestly, I also see it somehow. I cannot, you know, I know that it sounds even, you know, like spiritual, but I mean, it's a medium that is catching the information. Obviously.
Oh, come on, nothing spiritual. No, come on, no spirituality here. It's so obvious. You have a structure, you have a molecular structure that depends on the pressure,
yeah,
and the volume, and it will be measured, and it is measured, and that's why you get the high correlation with the volume. Yes, it's beautiful.
Interesting, so interesting.
Very nice.
Pierre, you have you raised hand. Pierre.
I'm sorry to interrupt again. Is it possible to share the screen or?
Okay, we have to ask Daniel, could you please just stop sharing your screen and
of course.
We can try to, Pierre, you could try to share yours.
Yeah, we'll try share the screen. Okay, where is it? Let me see, because this is an experiment that Daniel could very easily do. So let me see, we're sharing screen, where's the sharing chat? How do you share the screen?
Regarding the experiments that Daniel is suggesting, I just want to, while Pierre is looking for how to share the screen, I just wanted to say that as according to the description that Daniel gave me, it is incredibly easy to actually start growing Paramecia. So any of us can start doing experiments with Paramecia.
Absolutely.
we don't have to, you know, overwhelm Daniel with all our ideas of what and how he should do his experiments. He already has so many of his own things that he would like. Are you doing, Pierre?
Elena, I think
Yes, yes.
the host needs to give permission to share.
Okay, Lawrence. It is given. You have a permission, Pierre.
Have a permission? Okay.
You have.
So I'm not quite sure. Can you see the
Thank you,
screen?
Tomoko. Well, not yet.
What do you see? A PowerPoint presentation with origins as a title. Hello?
Not yet.
Not yet?
Yes.
Okay. Maybe I have to reshare it again. So let me see. Okay. So how about now? What do you see? Do you see anything?
No.
No.
Lawrence, is
Okay.
hearing okay? Is he? Are you using a Mac?
Yes.
Yes, he does. He uses Mac. You have to do some security settings in Mac in order. So that's the
Makes it
creating
messy.
problem.
Yeah. It makes it very, very messy.
It's very messy.
Okay. No, let's do it this way. I put in the chat a reference,
Okay.
which is
I saw
Kudakov
it.
et al. I'm not quite sure. So it's Kudakov et al., which is a paper published in 2011.
So,
1264 nanometer, right?
Yes, yes, oxygen dependent.
1264. Yeah.
And
Okay.
what did these guys do? So they used an infrared laser with 1260 nanometers, so in the near infrared range, and illuminated a sample of 10 milliliters of water. So the illumination took place for 5 minutes only. And they measured at the same time the biphoton emissions. Nothing happens for the next half an hour or so, 2 hours or so. However, afterwards, an underlying chain reaction obviously augmented the effect. So the biphoton emission started to increase by 3-fold. So the laser pumping activity in the near infrared, even if it's energetically lower, induced an emission in the UV range because they used filters. They could see that the chain reaction, as postulated by the water chain reactions postulated by Emilio Del Giudice and Vladimir Voyekov, is pumping up the water in a coherent fashion as to emit higher energy in the UV range, even though you illuminate it with infrared.
Infrared, yes.
1260 nanometer infrared.
And
So
this is
Daniel. Yeah.
This is singlet oxygen, by the way.
Yes, single
1264
triple oxygen. Yes.
is a singlet oxygen, yes.
Yeah. And if Daniel would use just one inducer colony and test it, what happens if he illuminates it with a laser light? If the population inside the inducer colony, without using the tester colony,
To
changes.
start produce.
I see the point.
Okay.
I see the point. I got it.
It's a
You
good
can send me the
homework for
paper.
collaboration. It's a very good homework for collaboration.
Definitely.
Yeah. Amazing.
Okay, well, guys, let's see if there is let's say one last question, because we are again extended this webinar as usually half an hour longer than it should be. It was quite interesting.
Well, it's worth. Yeah.
So last chance, any comments, any question for Daniel? Daniel, do you have any comments? Some last words.
I just greatly enjoyed listening to all of you, because it just enlarges the mind, the way to look at the world, you know, and it gives me a very, very good feeling that kind of my dreams I had years ago, you know, they kind of become real. Other people become interested in that phenomenon. and I become interested in the phenomenons they observe.
Yes, 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 yes.
This is all wonderful.
I completely have the same feeling when I listened to your presentation. I was like, and I actually, these are the works that I like best. I'm listening to one talk, but it gives me like 6 different directions in which now I would love to go and search more. So it gets me really inspired. And, you know, I really admire you for staying calm while you talk, because I really, you know, like when you make a emphasis on, and I just want to say, I'm only interested if there is an effect. I'm not looking at the causes. And like, I would die to know the cause. Oh my God. You know, and this is really what, I mean, this is what is a major inspiration for me. I would love now to know all this, what is the cause of the effects that you observe, and to try to explain them, you know, starting from the nano level, because everything starts there, in my opinion.
I agree. You know, Jelena, I have to learn to force myself not to say more than I can based on the data I receive.
Yes,
Absolutely.
I understand. Yes,
Yeah.
yes, yes.
And this is why I talk like that, you know, to give a maximum of clarity to you when you listen to something which is quite new, that you get it as close to what it really is. Or
yes, and that makes you an excellent scientist, actually. We should all be a little bit cautious with what we say. Yeah. Well, then, unfortunately, I think this is it for now. I wish we have more time. And I would like to say, Daniel, thank you so much for accepting to do this webinar. I'm glad you were here, and I hope we can, you know, like do some work together in future. Thank you all for attending, and especially, oh, this is very nice comment, beginning of the new world of science. Oh, what a beautiful, beautiful comment. I don't know who is this.
What is NDPC?
Yes, I don't know the name. I mean, oh, Tomoko-san, thank you so much.
Okay.
Who is NDPC? Can you please just, you know, like let us, wow, my countrymen. Oh, Dr. Nebojša Doveženski. I saw there are 2 people coming from my country, Professor Ivanka Karadžić, and this is a new, I don't know, but I'm so honored that you came to the webinar. It's so nice to see you. Let's talk more. Not new. I didn't see you before at the webinar. Because you're NDPC. You have to please put your name so we would know who you are. Thank you for coming.
Okay.
Thank you. Very nice to see you all. So we see each other again in a month. We will have another webinar with a new topic. And I guess this is it for now. Send us your, so the webinar is recorded. In few days it will be on the YouTube channel. If you had any other comments, questions, please feel free to interact. You can post your comments. You can contact Daniel. On our Aquaphotomics website, we already have one like a preview video lecture of what Daniel talked about, and his really beautiful paper, or as he calls it, a small book. So there are many ways, you know, to continue this talk further. So that would be it from me, I guess. Professor Zenko.
I just want to, I saw in the chat the very, some, I don't remember the name, Remy.
Remy, yes.
He was interested in the software, which is uploaded at our website, aquaphotomics.com. And this is something that could be used. And he gave a very good idea. I don't know if Zoltan is listening now, but we can devote some of our webinars to how to use the software. So Manuel, we have been thinking about this, and that was a good, also in the chat, a good idea. Daniel, I just want to thank you for absolutely a manual kind of a lecture, not so informative, at the same time scientific and showing how we can go further, step by step. So we cannot do so many things, but you showed exactly the points which, if we start with, it will start opening up. And to me, it was eye-opening, and I think it told me that, and with so many people today attending the lecture from different areas also, it is a collaboration. This is what we need. Aquaphotomics is a fruit of collaboration and interdisciplinarity, and this is our age. I mean, this is the time of multidisciplinarity and collaboration. I think this gives a very bright, I think, future, that this is the way. So I hope that in the future we can continue together. Thank you very much.
Thank you too. All the best to all of you.
And thank you very much to everybody attending the webinar. I'm sure that you found it very, very interesting. And so we're expecting the next one, Antonella, I think, right, Yelena?
No, the next is Professor Ozaki. We will soon send the announcement, and then after him is
okay, sorry.
So we will have more interesting webinars coming. So thank you all for coming today, and especially to people who attended this webinar from United States in the middle of the night. I'm so happy to see you. I'm sorry that we cannot make this available in all time zones somehow to be real time. We don't have yet the solution. It was very, very nice discussion.
Thank you.
Thank you all. See you in 1 month. Bye-bye.
Bye-bye.
Bye-bye.